Here is the overview of electron transport chain. Before I get into this, I'll ask you a couple of questions. Why do we eat food? To get energy. What is the energy? ATP, right? See, this is what happens. That's it. Look at the figure. When you eat food, so you eat carbohydrates, proteins, and lipids. Of course, you need vitamins, minerals, because it will aid in the metabolic process. So when we eat carbohydrate, proteins, and lipids, yesterday you have seen they will break down into simple molecules. Carbohydrate breaks into glucose, proteins breaks into amino acids, lipids breaks into fatty acid. These are simple things. And they will undergo oxidation in our cells. Just to go towards energy formation, these simple things will undergo oxidation. Glucose undergo oxidation, glycolysis, TCA cycle, you get NADH, FADH out of it. Similarly, fatty acid undergo oxidation, again give you acetyl-CoA, TCA cycle, FADH, NADH. Amino acids also can undergo oxidation if there is a need for energy. So out of 20 amino acids, you have seen already in that self-study lecture, out of 20 amino acids, 18 of them are glucogenic. Remember the small little table? 2 amino acids are ketogenic, leucine-lysine, ketogenic, leucine-lysine. Other than these two, all other amino acids are glucogenic. That means you can make glucose out of it. And that glucose goes into oxidation. Ketone bodies also oxidized. So eventually they will also provide a TCA cycle intermediate, that is acetyl-CoA. As you see, acetyl-CoA is a final common intermediate in all our metabolic pathways, especially in oxidation process. If your food is undergoing oxidation, macromolecules in the food, if it is undergoing oxidation, the final common metabolic intermediate is acetyl-CoA. Remember, and that acetyl-CoA undergoes citric acid cycle, which is a TCA cycle. The outcome of TCA cycle is what? You get NADH and FADH. NADH plus H plus FADH2. That's how we say it. So whether it is NADH plus or FADH2, you get 2 protons. And these protons systematically transferred through different complexes in the electron transport chain. And ultimately, these 2 protons, they will combine with an atom of oxygen, not oxygen molecule. Oxygen molecule is O2, 2 atoms. Together is O2, right? So this 2H is going to combine only with 1 atom of oxygen in the final complex of electron transport chain and make water. As you see there, 2 protons combine with an atom of oxygen and make water molecule, H2O. And that is what happens. And then, again, here in TCA cycle, not only 2 protons are released, you also get 2 carbon dioxide for every acetyl-CoA entering in. That means there is formation of water, there is formation of carbon dioxide when your macromolecules are oxidized. Whether it is fat, carbohydrate, protein, when it is oxidized to acetyl-CoA, further it is oxidized to, release carbon dioxide, and in return it is giving protons. Those protons are going into electron transport chain, ultimately combining with oxygen atom to make water. So what is the overall outcome of this? The overall outcome is, as these protons are going from one complex to another complex in the electron transport chain, the energy present in the protons is systematically used to convert ADP with PI to make ATP. ADP combined with PI, that's an endergonic reaction. If you are combining 2 things together, you need little energy, right? From where it will get, from where this electron, mitochondria gets energy. It will get energy from the flow of protons. As the proton flowing from one complex to another complex, it start releasing energy. That energy is used to combine ADP with PI to make ATP. And that means your electron transport chain is generating ATP when it is oxidizing the macromolecules. When cell is oxidizing macromolecules, ultimately what the cell will get is ATP. And what is the byproduct of this ATP product formation? It is carbon dioxide and water, right? Carbon dioxide, what we do? We exhale it. We just exhale the carbon dioxide and we inhale oxygen. See that oxygen which is inhaled, it will go to electron transport chain and it will become water. Whereas we are just exhaling the carbon dioxide. So inhalation and exhalation. Which system? Respiratory system. So the electron transport chain is directly connected with the respiratory system. That's why electron transport chain is called as respiratory chain. See, it's written there. Do you see that? Electron transport chain, down there written respiratory chain. Why? Because our respiration is directly connected with electron transport chain. So whenever your ATP production is increasing, example, when you are exercising, you need more ATPs, right? When your ATP production increases, what will happen to your respiratory rate? Will also increase because there is a need for oxygen. If you want to produce more ATPs, you need to supply more oxygen. That means your respiratory rate has to increase. And that's what happens when we exercise or when you are in stressful situation. You know, when you are anxious, anxiety, respiratory rate increases. If someone is in, in that crisis, anxiety, acute anxiety, their respiratory rate increases. Why? Because ATP production. That's a stressful situation. So whenever there is increase in electron transport chain, respiratory rate increases because oxygen has to be inhaled, carbon dioxide has to be thrown out. Okay. So this is how the things are connected. And then electron transport chain is also referred as oxidative phosphorylation. Let me just explain that and then we will go into arrangement. So what is going on here with your macromolecules? What kind of metabolism going on? Metabolism can be divided into catabolism and anabolism. Catabolism is oxidation. Anabolism is synthesis. So what is going on here in the figure? Oxidation, catabolism, right? Things are oxidized. When you are oxidizing something, so you are getting those two protons, right? Upon oxidation of the major macromolecules, you are releasing the protons and of course carbon dioxide. And those protons go through electron transport chain. And as they go through electron transport chain, what is happening with the ADP here? Down there, ADP has to combine with PI to make ATP. That is what is called phosphorylation. You are phosphorylating ADP with PI to make ATP. This is phosphorylation. So that means this phosphorylation happens only when there is oxidation of your major food macromolecules. Is it not? Only when you are oxidizing something, then only you are able to phosphorylate ADP with PI to make ATP. Is it not? Only when the left side of the figure is going on, then only you can do the right side of the figure, right? They are kind of connected. When oxidation is there, you can conduct phosphorylation. Without it, you cannot. So these two processes are combined or connected. That is why electron transport chain is also referred as oxidative phosphorylation. That's another name for electron transport chain. So these are the alternate names that you need to remember. So whenever you come across electron transport chain, also remember electron transport chain can be written as oxidative phosphorylation. It can be written as respiratory chain. Is it not? Respiratory chain, oxidative phosphorylation, electrons. These are all same. So make sure to know those terminologies. Coming to the concept, how much of energy that is stored in the molecule is actually captured by electron transport chain to make ATP. Like if you take an example of glucose, the amount of energy that is there in a single molecule of glucose, in that, how much percentage of energy can actually be tapped by the electron transport chain to make ATP. If you look at this figure here, and also I have written down here. So out of the 100% of energy that glucose has, electron transport chain can only capture 48% of that energy. Out of 100%, we can only tap 48% of total energy present in glucose to make ATPs. Rest 52% of the energy, 52%, it is simply released as heat. When you break a molecule, when you do oxidation, it's an exergonic process. You are breaking something. When you break a molecule, it releases energy. Previously I asked, when you combine ADP with PI, it's an endergonic process. Endergonic, you are combining. That means you need to provide energy to combine things. When you break it, it releases energy, right? So here you are oxidizing glucose into carbon dioxide and water. That means it is releasing energy. In that, 48% is converted to ATP, 52% released as heat. So that 48%, which is converted to energy, is 32 ATPs. 48% of energy coming from glucose is converted as, means used to make 32 ATPs. Okay, so rest is all released as heat as it is shown there. So my question is this heat is useful or waste? Did we waste it, or is in any way it is useful? It is useful. Why? It keeps you warm, correct. It is useful because it keeps you warm. That means it is going to maintain. We could put your, if I'm walking here, right, I might, no, it's fine. I'll be careful because you're tall and otherwise you, I will be careful. Yeah. See, there is a shadow, so I'll maintain the shadow there. I won't go beyond it. So what I was saying was, the heat that is released, it will maintain our body temperature, and that is 37 degrees centigrade. The room temperature right now, it may be 24, 25 degrees, right? But our body temperature is 37. That means 12 degrees higher than the outer temperature environment, right? That means how our body maintain 12 degrees higher than the external temperature. It's because in our body, metabolism is going on. Every cell is oxidizing these molecules. And when there is an oxidation going on, there is a release of heat, and that maintains 37 degrees, which is an optimum temperature for our enzymes. Remember enzymes, that bell-shaped curve, 37 degrees. So that is what is heat here. And also, when you eat food, don't you feel little warm? And also some people start sweating actually when they eat. Do you have any friend who sweats when they eat? Some people start sweating when they start eating, right? And also as such, when we eat, like maybe in a couple of minutes or during that time or a couple of minutes later, you feel a bit warm. You don't see it actually, but it happens. It's because that is called thermodynamic action of food, because your intestine is contracting. It has to, start digesting. A lot of ATPs are being used. When ATP breaks into ADP, the terminal phosphate, when you break it, there will be it's an exergonic reaction. ATPs are energy is released when you break ATP, right? So that makes the temperature bit higher. Okay, so overall, whatever heat that is released, that's a useful thing. Coming to organization of electron transport chain. All these complexes that you are seeing here, it is written as complex 1, complex 2, complex 3, and complex 4. They are all present in the inner mitochondrial membrane. Okay. So only thing is complex 1, 3 and 4, complex 1, 3 and 4, they are embedded in the inner mitochondrial membrane. Complex 1, 3 and 4, they are totally embedded across the inner mitochondrial membrane, right? Whereas complex 2, it is present towards the inner side of inner mitochondrial membrane. Inner side means matrix side. Complex 2 is kind of projected into the matrix while attaching to the inner mitochondrial membrane. If you see, like if I write this side as matrix of mitochondria, and this is inner mitochondrial membrane, okay. So complex 2 is kind of projected into the matrix because it is also part of TCA cycle. Complex 2 in the electron transport chain, it is also it acts as one of the enzyme in TCA cycle. We are going to see that. Complex 2 is, it also participates in TCA cycle. We are going to study this, what exactly the name of the complex 2 which participates in TCA cycle. So that's why it is projected into the matrix, while on the other side it is also participating in the electron transport chain. Other than this complex, other 3 complexes which are fixed complexes, complex 1, complex 3 and complex 4, they are embedded in the inner mitochondrial membrane. Now, these are protein complexes, complex 1, 2, 3 and 4. There are 2 complexes in the electron transport chain which are referred as mobile complexes. Mobile in the sense they move from one place to another place. Within the inner mitochondrial membrane, as it is shown in the figure, there is Q and cytochrome C. And this Q here, generally, in most of the books, in this figure it is written Q. Otherwise, it is generally written as CoQ. CoQ. CoQ is coenzyme Q. Coenzyme Q. Remember CoA? CoA studied? Coenzyme A. In vitamins, did you see somewhere coenzyme A? Which vitamin is essential to make coenzyme A, CoA? B5, pantothenic acid. That's the only function of B5, to be part of coenzyme A. Okay, in the same way, this terminology here, CoQ, coenzyme Q, CoQ. Okay, this CoQ is a mobile complex, which is, you got to know the location, where it is located, because that is important for understanding purpose. So it is located between complex 1, complex 2 and complex 3. That's where it is located. Okay, remember that. And then cytochrome C. Cytochrome C is located between complex 3 and complex 4. And this is also mobile complex. So there are 2 mobile complexes and 4 fixed complexes in the electron transport chain. That's how it is organized. Okay, 2 mobile and 4 fixed. All right. So this is how anatomically your electron transport chain is organized. So now let me take you into, yes. No, it is in between complex 2, 1 and 3 as here. That's a separate complex called mobile complex. No, 1, 2, 3, 4. And then 2 mobile complexes. These are fixed one. Fixed complexes are 4, 2 mobile complexes. The 5th one is ATP synthase. I'll mention it later. Okay. So, is it clear? Arrangement. Okay. So, especially you need to know the location of CoQ and cytochrome C, because they will go back and forth. Because they are mobile, they move from one place to another place, and we need to know the location so that we understand what exactly it does later. They move from it. They just like act like a courier guy. You know, they go to collect from the source and give it to the wherever it has to go, right? So back and forth. So that's something like that. Anyway, we'll get into that. Coming to the flow of electrons in the electron transport chain. How exactly electrons flow in these complexes? So this figure looks a bit complicated because the way it is written, but I'll try to explain and simplify it as much as possible. Okay. So before I start explaining you the flow of electrons in the electron transport chain, you got to remember one thing here. The way electrons flow from one complex to another complex, it all depends on negative redox potential. Say, let's say redox potential. The principle here is redox potential. Redox potential. What kind of redox potential? So the flow of electron, it will go from negative redox potential, negative redox potential. I'm writing that down here. Negative redox potential. It starts from here, and that means NADH plus has got highest negative redox potential, and it will flow all the way like this to oxygen. It goes all the way like this to oxygen, and this oxygen has highest positive redox potential. You don't need to remember exact number for redox potential, although briefly I have written that in the notes below. Those who are using, did I write redox potential numbers down? No, did I write notes? I didn't mention anything. Sorry. Which slide I have written? Somewhere I have written redox potential. Did I write? Okay. So anyway, my point here is, in general, what you need to know is this entire electron transport chain is designed based on redox potential. That's the first thing you got to know. Okay, what kind of redox potential? From where to where they flow? Always remember, any molecule that has got highest negative redox potential, highest negative means negative number, like say minus 0.8 or something, right? And going into highest positive redox potential. Highest positive redox potential in the sense, say, plus 1.2 or something. So when the number go from negative all the way towards positive, so negative redox potential to the positive redox potential, that's how they flow. Okay. Now, any molecule that has got higher negative redox potential, it has got an ability to donate electrons. They donate. Molecule having a negative redox potential, they donate electrons. And the molecule that has got positive redox potential, they act as an acceptor. So donor and acceptor here. So higher negative redox potential means act as a donor. Positive redox potential is acting as a acceptor as you go from higher negative to positive number. Okay. Now, with that concept, if you look at this figure, it starts from NADH. Look at here, NADH. That's where it got to start reading this figure, NADH. And this NADH is going to donate its electrons and protons to complex 1. And that complex 1 will donate its electrons and protons to coenzyme Q, because that's the next complex, that's the mobile complex. So coenzyme Q come here, collect electrons, collect protons, and then go to complex 3 and give it. So complex 3 will give its electrons and protons to cytochrome C, because cytochrome C will come and collect that, because that's a mobile complex. Cytochrome C will go and give its electrons and protons to complex 4, and the complex 4 finally will give the electrons and protons to oxygen atom. So that means final acceptor of your protons, that is 2H, is oxygen atom. And oxygen atom, after accepting those protons, become water. So the final acceptor is oxygen atom, and the initial donor is NADH. In between, these complexes will go towards positive side. From negative side, they go towards positive side. Okay. Just to make this little more clear, we will use random numbers. Don't write it down. This is just for example and you don't need to memorize. Just random numbers I'm going to write. Okay. So what I'll do now is don't write it down. Okay. Otherwise you get confused when you study yourself. Then you start remembering numbers. Don't get into that. It's just understanding the concept. So I'm going to write minus 0.8 here. Minus 0. Remember, minus negative redox potential. Okay. And then complex 1, I'm going to write minus 0.6. Okay. And coenzyme Q, I'm going to write this coenzyme Q here. Minus 0.5. And then complex 3, I'm going to write minus 0.2. And then cytochrome C, I'll write plus 0.1. So I'm going towards plus now. And then complex 4, I'm going to write plus 0.2. And then oxygen, I'll write plus 0.3. Now see what happened to your molecule and going from negative towards positive side. Is it not? So as your number go from higher negative side towards higher positive side, they start becoming an acceptor of electron rather than the donor. So now let's look at this here. NADH plus has got minus 0.8. So it is acting as a donor of its electrons to complex 1 because complex 1 negative redox potential is little less than the than the NADH. So that means complex 1 can accept electrons from NADH plus, and then that complex 1 can donate its electrons to coenzyme Q because it has got little less negative redox potential than it. And then like that, each one of them, they accept from previous and he is able to donate to the next. What's your name? Paola. Paola has $1,000. What's your name? Ariana has $800. You have $400. Not 600. Marco has 400. Arib has 200. Sujay has $100. Who has got a higher ability to donate? Paola can donate. Can Sujay donate to Paola? No. Always rich guy has to donate, not the poor guy. No, I know you are rich, but is it not? See, the ability to donation will come only when you have little more than what you need. Is it not? If you donate when you don't have, you will depend on somebody else and you will beg from them. You should not do that actually. If you don't have, in fact, it is good not to give. It means you don't be too greedy. But like, first thing is we'll have to look after ourselves, right? Before we give it to others, right? So we get an ability to donate only when we have more. So molecule that has got higher negative redox potential, it has got an ability to donate because it has more. And then the next one, like Paola can give it to Adriana. Adriana can receive from Paola, right? She cannot receive it from her. Sorry, I forgot your name. Because she has little less than Adriana. It's like that. It cannot go in this way. It can only come from that side. So in the same sense, in the electron transport chain, it can only flow in that direction. Okay. Now I will introduce one more thing. So FADH2. So there is another molecule called FADH2. This FADH2 has got a negative redox potential of, let's say, just for example, 0.55. It is all measured in volts or millivolts. This is electric potential, negative redox potential. Okay, so this FADH2, can it enter into complex 1 and give electrons and protons? No. Why? Because complex 1 is minus 0.6. It cannot act as an acceptor from FADH2, but it can act as an acceptor from NADH. Whereas FADH2 can donate its electrons to complex 2 because it has minus 0.5, which is little more towards positive than its own. Is it not? This is how the flow of electron goes, and this is the basic principle of electron transport chain. That is why the flow is very systematic. They have to go in this direction. Okay, so that is the redox potential. So what you really need to remember is, it is electrons and the protons in the electron transport chain, they flow from highest negative redox potential to highest positive redox potential. That is the point. That's what I think I have written down there. Is it clear? Okay. Okay. So with this understanding, now let's move on to see what happens to NADH and what happens to FADH, how exactly they flow all and go all the way to oxygen atom. So we have complex 1 here, and then coenzyme Q. There are 2 coenzyme Qs shown here. That is where you may have confusion, right? So just to take out your confusion, what you need to do is you just follow the arrow here, big arrow. This big arrow is taking you to this coenzyme Q. Okay. And then the coenzyme Q is reduced to coenzyme QH2, and then that it gives it to complex 3, and then after that, you know, it is flow is okay. Second confusion is complex 2. Complex 2. Depending on the cell type, depending on which tissue you are looking at electron transport chain, so there can be 3 types of complex 2. 3 types of complex 2. Right? 3 types in the sense, we have succinate dehydrogenase, which is part of TCA cycle. That is also complex 2. Then we have ETF, electron transferring flavoprotein. We are going to go into detail of that later. It is participating in beta oxidation of fatty acid. Beta oxidation of fatty acid oxidation. That's where you get ETF, electron transporting flavoprotein. That's also complex 2. And then we have something called as glycerol 3-phosphate dehydrogenase enzyme in glycerol phosphate shuttle mechanism. We're going to study this in glycolysis. Glycerol phosphate shuttle in glycolysis. And that is also acting as complex 2. So that means depending on the tissue type, depending on the requirement of the cell, depending on which metabolic pathway you are looking at, how they giving their electrons and protons to electron transport chain. So considering all that, we have 3 complex 2s. Okay, and that is succinate dehydrogenase, electron transferring flavoprotein, and glycerol 3-phosphate dehydrogenase. All of them, they receive their protons from FADH2, as I shown earlier. FADH2 will enter into complex 2, either of these complexes, any of these 3 complexes, complex 2s, any of these complex 2s. FADH2 enter. So, and then they will give away their electrons and protons to coenzyme Q, and then that will give to complex 3 and then move on. It's like that. Okay. So they wanted to show complex 2, how they their electrons and protons will flow, and also complex 1, how their electrons and protons flow. That is why they have written coenzyme Q 2 times. Otherwise, if I go back to this figure and make it simple, keep it simple. Let's use, say, this complex 2, succinate dehydrogenase, right? So FADH2 coming from here, FADH2 entering here and donating protons here, H2 given here, and they simply go to complex 3 from here, and then cytochrome C, complex 4, and then oxygen atom. It just flows that way. That means your FADH2 always enters into complex 2. That's it. It never goes to complex 1. It just starts with complex 2. With this figure, you can understand. Imagine instead of succinate dehydrogenase as complex 2, instead of that you put glycerol 3-phosphate dehydrogenase. Flow is same. Or you put ETF here. In place of succinate dehydrogenase, you put ETF. It is also same. So they just wanted to show everything in one place in the next figure. That's why figure is bit complicated. You got the idea? Yes. Coenzyme Q is same guy, receiving from everybody. Coenzyme Q is the same molecule, just collects from everybody. Okay, it's like you have a delivery guy, 3 of you, neighbors, and I am the delivery guy. I collect package from you, and give it to him. That's it. I am coenzyme Q. You 3 are 3 complex 2s. All of them are S2 students. Complex 2s. Same. Is it not? You got the idea? So it is just that there are 3 complex 2s. Complex 2, there are 3 of them. Depending on which type it is, what is the metabolic activity of that cell, where you are looking at. If you're looking at TCA cycle, your complex 2 is succinate dehydrogenase. If you're looking at beta oxidation of fatty acid, your complex 2 is beta oxidation of fatty acid. What is your complex 2? ETF, electron transferring flavoprotein. Remember flavoprotein, riboflavin. Okay. And then if you are looking at shuttle mechanism, glycolysis, your complex 2 is glycerol phosphate dehydrogenase. So it all depends which metabolic pathway you are referring to, and that is the complex 2. Okay, is it clear? So complex 2 will give its electrons and protons to which mobile complex? Coenzyme Q. And then coenzyme Q will carry it to complex 3. Complex 3 will carry it to cytochrome C. And cytochrome C will carry it to complex 4 and then ultimately to oxygen atom. So because these complexes, that is complex 1, complex 3, complex 1, 2, 3 and 4, as you see here, they are fixed complexes. You know, they are fixed to the membrane. They cannot go. They cannot move to another complex and offload content. They need somebody to come and collect and give. Who is going to do that? CoQ. As you see here, CoQ is a mobile complex. Why it is called mobile? Because it can move. This CoQ, it can move. Within that membrane, it will go here, collect, it will go here and give off, something like that. So this CoQ, it will move back and forth. So it means it will come to complex 1, collect those protons, and then go to complex 3 and give it. In the same way, it will go to complex 2, collect, and give it to complex 3. It is just like it moves back and forth as a delivery guy. So if it is a coenzyme Q can move within the inner mitochondrial membrane, which is entirely lipid, what kind of structural arrangement this CoQ should have? Within that lipid, hydrophobic environment, it can easily move here and there. So it should be hydrophobic, right? It should be hydrophobic so that it will be soluble there and it can move from here and there. It's like that. You're getting the point. So mobile complexes are hydrophobic because they are there within the membrane. Similarly, cytochrome C is also the way it is arranged. It's a protein. The way it is arranged, it's hydrophobic. Outer surface is hydrophobic, inner core is hydrophilic. That is how they are built. So CoQ is a lipid molecule, whereas cytochrome C is a protein molecule. Both of them, they retain the hydrophobicity. Okay, that is why they are able to move from place to place. Is this clear? Complex to complex, correct. They are the ones who will move electrons and sometimes protons from complex to complex, one complex to another complex. Any doubt? You? What's your name? Alexa. Easy to remember. Anytime say Alexa, sing a song. At that time there was Alexa, some box. Nowadays, not many people use that. I don't know. What is whether like today and some couple of 10 years back, it was famous. Alexa, yes. Coenzyme Q, yes. It will accept from complex 1 and donate to complex 3. Accept from complex 2, donate to complex 3. Yes. Okay. So, this is what, yes. So, ETF, just remember as ETF. ETF is electron transferring flavoprotein. Electron transferring. Transfer. Let's say transfer. Actually, it is transferring, but it's okay. Transfer. Electron transfer. Flavoprotein. It is made using riboflavin. That's why flavoprotein. Any name flavo, it is always riboflavin. Derivatives of riboflavin. Okay. So these are 3 complex 2s. Succinate dehydrogenase, electron transfer flavoprotein, which is there in beta oxidation, and glycerol phosphate dehydrogenase. So they will give their electrons and protons to coenzyme Q, as it is shown in this figure. See the arrows, they all will give it to coenzyme Q, which become coenzyme QH2, and then it will go to give it to complex 3, and then cytochrome C, and then complex 4, finally to oxygen atom. So just to finish this, the last concept. Tomorrow I will continue with the other slide and then do the overview and see. We will see how ATPs are synthesized and understand inhibitors and uncouplers after the quiz. Okay. Now the thing is to look at some of the components of these electron transport chain complexes, because the names for each one of them is based on that. Sometimes it is based on the function of the complex or based on the content within the complex. If you carefully observe this figure here, especially focus on the fixed complexes, look at complex 1. It has FMN. What is FMN? Flavin mononucleotide. Fe is iron, S is sulfur, iron-sulfur cluster. So the complex 1 has FMN, iron-sulfur cluster, and what it does is, it is going to receive these electrons and protons from NADH, and then it is going to give it to coenzyme Q, right? That's why look at the name of this complex. It is NADH, CoQ, oxidoreductase. It's going to oxidize NADH into NAD+. That's an oxidation process. And then what it does, it's going to reduce CoQ into CoQH2, because it's going to donate protons to that particular mobile complex, right? So it will take it from NADH while oxidizing NADH into NAD, and then it is going to give it to coenzyme Q while reducing coenzyme Q into coenzyme QH2, as you can see in the figure. And that's why name of the enzyme, this complex is called NADH CoQ oxidoreductase. Some authors simply write NADH dehydrogenase or NADH oxidoreductase. Sometimes they write NADH dehydrogenase for complex 1. Why dehydrogenase? Dehydrogenases, they belong to oxidoreductase class of enzymes. Remember, there is a self-study slide. I don't know whether you went through, you got to. There is a self-study classification of enzymes. Oxidoreductase class is very first, and the last name dehydrogenase. Dehydrogenases belong to oxidoreductase. That means complex 1 can be simply written as NADH dehydrogenase. Okay, let me write it down. You also write it down in case, so that you don't have any confusion later. NADH dehydrogenase. Dehydrogenase. That's the complex one. NADH CoQ oxidoreductase or NADH dehydrogenase, complex one. Coming to CoQ. CoQ, as you see here, it can be reduced to CoQH2, CoQ converted to CoQH2, which is oxidized and which is reduced in this. CoQ is oxidized, CoQH2 is reduced. So CoQ, which is a coenzyme Q, it is written as ubiquinone. Ubiquinone. Make sure you write it down. CoQ is ubiquinone. And CoQH2 is called as ubiquinol. Ubiquinol. Reduced form. Ubiquinone, ubiquinol. What I have written here is ubiquinone. That is CoQ. Ubiquinol, CoQH2. NOL, quinol. Okay, reduced form. So your mobile complex will go from oxidized to reduced back and forth. When it gets, it is reduced. When it is giving away, oxidized. So it is back and forth. That is your complex, mobile complex there. And there are 3 complex 2s, which we have already explained. So I explained it. So those are the names. Coming to complex 3. Look at complex 3, especially the content in complex 3. There is cytochrome B inside. There is iron sulfur cluster and cytochrome C1. See, there is cytochrome B and cytochrome C1 in complex 3. That's why name of complex 3 down here, it is written as cytochrome BC1 complex. Complex 3 is referred as cytochrome BC1 complex. Okay. And then we have cytochrome C as a mobile complex. It is just cytochrome C, mobile complex. Coming to complex 4, look at complex 4. In complex 4, what is happening? What is happening in complex 4? Like, who is going to donate these electrons and protons to complex 4? Cytochrome C. When cytochrome C is donating, means offloading its electrons and protons to complex 4, what is happening to cytochrome C? Whether it is oxidized or reduced? It is oxidized, right? It is oxidized. Who is going to oxidize that? It is the complex 4 which is oxidizing it by taking it from it. And that's why complex 4 is called as cytochrome C oxidase. See the name? It all explains the function. Complex 4 is oxidizing your cytochrome C by taking its electrons and protons. That's why it is called cytochrome C oxidase. And then some authors, instead of using the cytochrome C oxidase name, they will write this as cytochrome AA3. Cytochrome AA3 complex. Why it is written cytochrome AA3? Just like your complex 3, it is written cytochrome BC1 complex because there is cytochrome B and cytochrome C inside, right? In the same way, complex 4 has cytochrome A and cytochrome A3. That's why it is written as cytochrome AA3 complex. These are the names you need to remember. Why? Because when we make questions on the electron transport chain, inhibitors, uncouplers, mechanism, which one of the following, like riboflavin we connect, niacin we going to connect, right? And then ask which one of the following complex is affected by this. And then instead of what, sometimes we write complex 1, 2, 3, 4, 5, A, B, C, D, E. Easy. Some other time, some authors, even me, what we do, we write NADH CoQ oxidoreductase, then succinate dehydrogenase, then I write cytochrome BC1 complex, cytochrome A3 complex, ATP synthase, 1, 2, 3, 4. And then If you're looking, you're thinking, okay, complex 3 is the answer. My answer is complex 3 and looking for complex 3. The complex 3 not written. Somehow differently it is written. So complex 3 is what? Cytochrome BC1 complex. It is there in the choice. And that's where it will be useful. Okay, to identify the complex names in the choice. This is necessary. Okay, that's why you need to make sure the alternate names for complex 1, 2, 3, 4. These are the alternate names. So make sure you understand that, write it down, go over. Okay, so that's all for now.